I really enjoy the Open Talks meetings because you meet so many different people, the sort of people with expertise that I don't normally come across. And when Barry said, "Would you like to talk at the meeting?" I thought for a minute and thought, yeah, I remember the way I went away from the meeting in Athens. I went away with my head absolutely buzzing with a conundrum, and I thought it would be useful to share with you what it was that was making me so anxious and excited all at the same time, and also to think about ways in which we might move our way forward. Let's see if I can press the right button. So, when you're doing a talk, you always have to look up your term. So I looked up conundrum, and I thought that was actually quite a boring definition. So I kept Googling, and I found another one which I liked better. A riddle, or a riddle whose answer is or involves a pun, fine. A question or a problem having only a conjectural answer, that could be what we're trying to do. An intricate and difficult problem with a whimsical solution. So I thought, well, that's interesting. I wonder where that might take us. So what I've done is to try to define what I think the conundrum is in my terms, then try to analyse what I think the pieces are within that conundrum, some thoughts on how the problem will be solved, and I think we've actually heard a hell of a lot of the elements of how the problem will be solved in the last 2 days. And then finally a whimsical solution, which I hope isn't too whimsical, but it may be a step too far, but it might at least get us thinking. So what is the conundrum? And I went back into my records and I discovered a presentation that I was a co-author on at the Society of Toxicology meeting in 1989, which was initial invalidation of the in vitro technique for the assessment of skin irritant chemicals. So I've been in this game a long time. And when I went and checked up on it, I noted that that had become an OECD method. And it had taken a quarter of a century to get there. So I thought, well, okay, now things are different now, aren't they? And there'll be lots of other things that are sorted. And ontologies, classification, categorization. This is something from the TTT roadmap in toxicology that they came up with. It was just a way of kind of thinking our way through it. So we've got acute local, acute systemic, sensitization, mutagenicity, repeat dose, carcinogenicity, development and reprotox, and ADME. And I thought, well, let's look up and see what in vitro methods there are in the OECD in those categories. Oh, some big gaps, aren't there? Okay, we've got acute local, but if we haven't got acute local after 25 years, what the hell have we been doing? Mutagenicity, well, we've been doing Ames tests since 1970, goodness knows what. We've got estrogen agonist, steroidogenesis, and we've got some dermal absorption. But yeah, of course, there's lots in progress, isn't there? So I went and looked up the drafts. And we've got eye irritation and skin irritation. Skin sensitization is appearing. There's something about P450 induction, which I kind of thought, well, that's perhaps to do with carcinogenicity. And we've got something on the estrogen receptor, some even more refined mutation tests. So you can see we've still got big gaps. We haven't even got anything for acute systemic yet, never mind systemic toxicity, repeat dose toxicity. So my conundrum is, if it took 25 years to adopt alternative skin corresponding methods, what do we have to do to get alternatives in the more difficult areas before you're all dead, never mind me, and I'm a lot older than most of you. So I remember on the flight back from Greece actually scribbling down in my notebook all sorts of strange thoughts about what I thought was going on, and I'll share some of those with you, and I may upset a few people, but then that's the price of being on near the end. So the current paradigm is well, we've seen this before. It's not working all that well. It's stood us well. It's expensive. It takes a long time. At the moment, millions of euros are spent by agrochemical companies to develop chemicals. They they do tests to predict what's going to happen in the tests. And when they've done the tests, which they spend millions on, they then do more tests to try and work out what the results meant. I don't know whether is there a crisis?
Nothing to do with you.
Okay. So toxicology is spent 3 times: one to predict what's going to happen, once to find out, and then to understand it. That's wasteful. What are people worried about when we start to say, well, why don't we use the new methodology? Well, I'm overgeneralising. There's a lot of people in companies worry that the new methods are going to be even more sensitive than the old methods. The old methods have a lot of false positives, so the new ones will have even more. If you're in a green NGO, you go, well, these things aren't going to pick everything up. How can they possibly pick everything up? So they're worried. There's an interesting little group of anti-capitalist NGOs that actually see high costs in toxicology as a good tax on industry. So they're a bit worried if you come up with methods that are cheaper. And regulators have got a very sensible fear, which is they might approve a product which then turns out to be a real problem, and nobody wants to do that. Who else has got a stake in all this? Well, the academics are anxious as well, because they see great opportunities for grants and developing new methods and also creating new issues. I used to have a phrase for a chemical which was particularly difficult. You could call it a grantogen. And I've heard of at least $80 million, 80 million euros worth being spent since I've been here, 50 in the past and 30 to be spent in the next few years. And the consumer wants to be nice to animals, be safe from nasty chemicals, generally have a nice lifestyle. So can all these things be reconciled? Anything else? Well, millions of dollars have been invested in all this. We've heard today and previous days the efforts that are going on in the EPA, in the States, all the collaborative work that's going on in Europe. We're in the situation, as we just heard from Gladys, where if you're trying to develop new cosmetics, you can't actually use animals, but there's no tests that are approved. And we had this long and arduous harmonisation process, which has been great because it's labelled us all to know what we've got to do, but it has ossified the science in the 1980s. So who's going to sort all this lot out? That was what was really puzzling me on the way home from Greece. You know? Industry is waiting for the regulators to specify something. The regulators are waiting for the academics to develop something. The academics are all collaborating and saying my method's better than your method and this method doesn't quite fit with this, and everybody wants to work at a very detailed level. Nobody wants to seem to want to pull it together, with the exception of Michael and a few others. Meanwhile, we're paying millions for the tests that are currently being done, and we're using millions of animals. Does anybody have a plan? Will someone say, actually, we've had enough of all this now, we're going to do it like this? I can't see it coming yet, but it might do. The EU, interestingly, I'm going to offend Morris up there now, but seems to me it's outsourced approving to the OECD. If it's an OECD-approved method, we'll use it, but don't ask us to approve it. So is it going to be a bottom-up process where this group of people and others like it turn around one day and say, you know, I think we've got it? I worry about that. And even if we did do that, how do we stop ossification in the future? Because actually what we don't want to do is to inflict on our successors in 30 years' time what was good science in 2015, because in 2040 things will have moved on. So how might we solve this problem? Well, we've got a lot of things changing. The science and technology, we've heard an awful lot about that in the last few days. Well, a lot about that, an awful lot, a lot. We've also heard, especially in the States, there's this big political pressure on the number of untested chemicals that there are. There's a whole animal welfare issue. And we've got regulations that are building on that. So there's pressure and there's science going on. I think we're going to have to be more innovative. And a few years ago, the Medici effect was was fashionable, that it's bringing different things together to create something new. When I thought about that, I thought about biological science and computational science. But I think the third angle that we've got to work on, this is probably what my finish is going to be, is that it's a philosophy. We've got to ask the questions about human safety in a different way to be successful. So this is the regulatory acceptance process that a slide that I got from a seminar, a webinar that I went to, and you've seen it before. It's long, it's tedious, it takes 25 years to approve skin irritation, so I don't think that's going to work. REACH, ECHA are actually got their eyes open to the fact that there's new methodology, and they're laying down some guidance as to how you can bring in new methodology. You can see it's very much stuck in the old way of thinking. Results must be adequate for classification, must enable adequate risk assessment. Key parameters from the standard study are addressed, i.e. adequate exposure duration. Thoroughly documented scientific explanation, I can understand that, but the others are actually saying, please make it look as much like the old way as you can, which is slightly worrying. What we've started to see from a number of people, and especially that last slide, we've seen that slide with that clip of the number of non-specific compounds twice in this conference. And when I was a kind of a junior, wet behind the ears toxicologist, I asked my colleague at the time, who's now John Moores University, Ted Locke, when we were getting into neurotoxicity, what makes things neurotoxic? And he said, well, they've either got to get to the brain and be toxic, they've got to get to the brain and be turned into something toxic, they've got to overstimulate the nervous system, or they've got to inhibit the nervous system. And you go, hang on, yeah, that makes sense. And we're starting to hear that coming through as a way of thinking about compounds. They're either non-specific toxicants, depending on where they get to and where they end up, or they interfere with something more specific. So they're starting to get some clues. I'm not going to spend a lot of time on this. This is really basically coming back to the philosophy that we've heard already twice today and before, that if you start to look at what is it that starts to make chemicals more potent and more toxic, it's because they're either cytotoxic at low doses or they interfere specifically with processes, AOPs, whatever you want to call them. That's what accounts for, this is the right thing, yeah. This is the distribution of toxicity within the top rep database at Fraunhofer. And you can see, you've got things that are really toxic, really potent down at this end and not at the other. So how can we find ways to sort these groups out? Is it by understanding some of these parameters? So these are the things that make you more toxic, these are the things that make you less toxic. Starting to understand that, rather than just doing simple predictions. Once we've done that, we can start to put them into a framework. I'm very pleased that the EU has dropped 21 from the end of Tox Risk 21, because I was involved in Risk 21 and it would be very confusing. But this is simply says, actually start to use methods with low precision to start with, and then if you need to, get more and more precise. And you do exposure and you do toxicity at the same time. So if we do all that, will we meet these conditions of REACH? Well, I think we can do risk assessment. I think we can come up with ways of explaining how we do things in a way which are scientifically documented. How we relate it back to the standard studies, I still think there's an issue. And I think the whole issue of classification in Europe needs to be thought about very carefully as to how we're going to address that, but maybe more of that later. So moving into the future, get my crystal ball out, what do I think is actually going to happen? It's not very well standing here whinging about it. What do we think is going to happen? So I came up with a 4-phase process, and I was told last night it'd have to be 3, but never mind. So phase 1, you've got to do everything in animal. If you want to make a decision, if you want to make a regulatory or a business decision, you do it in animals. Phase 2 is you do animal studies by default, but non-animal based studies you can waive the need to do those studies if you've got non-animal based studies. So you've got the two things going on in parallel, but it's weighted towards animal studies. Then you move to phase 3, which switches round. So most of the time you do non-animal studies, and if you need to address any uncertainty that you've got, you then do animal studies, in vivo studies. And then phase 4 is non-animal studies, safety assessments using non-animal data, and no animal data are allowed. So that's, to me, there's an evolution through there. And when's this going to happen? I've got my other silver ball out, crystal ball out. And I think if you look at the menu there, I'm into impressions. You've got a $10 impression of the time I think this is going to take. So what I've done is to plot this out for four of the sectors. So if we follow general chemicals, we're actually in phase 2 at the moment, because you can do read across. You can come up with a good argument to avoid doing a 90-day study. You've got to put some good stuff in place, but we're there. I think that the use of that will generally increase in the next 5 years. So that by 2025, it may be more. It may be that actually you only do an animal study if you really need to. I've hedged my bets here. I think it may sort of work along the border. Some things like industrial or cleaning products will pop up. Others will stay down in phase 3. Pharma, I think, will lag behind. It's actually probably just on that cusp now. There's the harmonisation of carcinogen studies coming through. So I think that's going to follow along behind general chem. But I don't think it'll ever get into phase 4. I think people will always want reserve the right to get in to do some in vivo work if there's a particular issue. Agrochemicals, I think that will lag behind, because I think there are the political issues around agrochemicals that make people very nervous about moving, but it will happen. I think if it starts to lag behind general chemicals, I think the animal welfare lobby will say, "Oi, what's going on here? Why if it's good enough over here, why isn't it good enough over there?" Again, I think that will still always be the reserve to use an in vivo study, unless the politicians decide that they don't want it to be that way. Poor old personal care is in stage 4 already. So they've got a really difficult situation to manage, in that they've got nothing really approved. But as this catches up, personal care will be less out on a limb on its own. So I think that's my prediction of what's going to happen in the future. So how could we move things on a bit more from where they are now? Something interesting that Morris said earlier about the use of standard-based approval rather than a method-based approval. If we had a set of standards that we could put through any system that we wanted to demonstrate was good, then we could get into the crowdsourcing market and competitions. Why not have a competition which gives prizes to the methodology which meets the sets of standards? And what could that prize be? Well, ignore that. Opposite of what Morris said, why don't you make the prize be that actually you've got 5 years worth of monopoly for people to use that methodology? And then you'd have to repeat that every 5 years to encourage new developments and to prevent ossification. So as soon as competition 1 had been set, you then get into the next competition. Now, one of the things that's come to me and to others in the conversations during this conference around this is one of the things that I've learned when I investigated the use of some competitions in another context. And one of the things they drum into you when you talk to them is you've actually got to do your specification, and the way you set the problem out is really important. And I think it comes back to the conversation that Gladys and Morris were having just now about how are we going to present the results from this new way of working. And if you remember the paper we had from Tara Dalton about that elegant means of doing multiple PCRs in little drops, she set herself some criteria, which was we wanted a method that actually fitted in with the way people already work, and that they can actually just do it and use the equipment that they've got. And I think what we've got to say to ourselves is, can we come up with a way of describing the output from this which is consistent with the new methodology that we're developing and the new insights? And it's not just the methodology, it's the new insights. I mean, this breakdown between specific and non-specific chemicals, I think that's very important to start getting it through. Can we work away from, well, it affects the kidneys in a 90-day study in the low effect, low linear. But we've got to do that in such a way that we can present that to downstream users, risk managers and regulators in a way that makes sense to them and relates to the way that they want to give advice and to protect health. So I think to me, there's a piece of work that's come out of this, and I think, Maurice, you've already started doing some of this, which is, can we come up with a different, am I allowed to use the words ontology as a simple toxicologist, and different ontology about the way that we present our knowledge about the toxicity of a chemical. And that to me is what it's about. So, the conundrum. You remember the British Prime Minister used to have a famous phrase, there is no alternative. Well, I've changed that a bit. There are alternatives, but we need alternative ways of getting there as well as alternative methods. Thank you very much for your attention.